Hi, Miss Nikki here. Welcome to chapter 22. This is the immunity chapter. And just a heads up, we're doing more of an overview. We want to talk about the big picture, big process. What are we making? What types of cells are we making? How are we making them? What causes inflammation? What causes fever? But we're not getting into the very specifics of, CD4, CD8 receptors, or some of the chemicals that are being released. So I put this image up here because I just wanted you to see this cytotoxic T cell. Most of us could look at that right away and say cytotoxic, okay, well, it's probably going to kill cells, right? So these cytotoxic T cells, we'll talk about how they're generated along with helper T cells. And these come from lymphocytes. So if you remember the A granulocytes that we talked about, so we'll see kind of that process if you don't remember. Hopefully we can look at these 5 categories of pathogens or infectious agents. We see bacteria, virus, fungi. We may not be familiar with the bottom 2, like protozoans. It's a eukaryotic cell that doesn't have a cell wall. Plasmodium is the protozoan that causes malaria. It basically infects your blood, infects your red blood cells, and causes them to lyse. Multicellular parasites like tapeworms. Just to be clear, if you see the word pathogenic, that means it's causing some sort of damage to the host, right? But you also have good bacteria. So they might use the word microbe throughout this chapter. That's just bacteria can be a microbe. It's not pathogenic unless it causes you harm. And unfortunately, we only know the ones that cause us harm. So we kind of get this idea that all bacteria are bad. But if you ever seen any yogurt commercials, you know that there are good live culture bacteria that are probiotics. They help reduce inflammation. So don't think that they're all terrible for us, right? I don't have this term highlighted, but I did want to bring your attention to this term prion or prion. Basically, it's an infectious protein. It's a misfolded protein, and when it comes in contact with other proteins, it causes them to misfold. So there's no cells, there's no virus. It is basically an amino acid, protein, and it can cause nervous tissue disease. So mad cow disease is something you may or may not have heard of in the news. It can spread from cows, so these prions can spread from cows to humans if we consume infected meat. Not only do we have white blood cells in our bloodstream, right? So they give you the broad topic of leukocytes. So all of our white blood cells, do we have them floating in our blood? Yes. How do we create them? They're formed in the red bone marrow. Hopefully, we can go back to hemocytoblasts, and we know that those are going to make red blood cells, erythrocytes. They're going to make thrombocytes or platelets. And then they're also going to create white blood cells, right? We're going to talk about how they get differentiated at the white blood cell level in terms of creating T cells and B cells. So hopefully we remember something about here. We're going to have kind of this lymphocyte differentiation, and then we're going to have this monocyte. So for the lymphocytes, this is where we're going to see the T and the B, and the monocyte, this is where we're going to see the macrophage. And we'll look a little bit more at this. They also want you to realize that all of these leukocytes can be in lymphoid structures. So we kind of skipped ahead and we haven't talked about lymphatic system yet, but lymph nodes. So you have lymph nodes, cervical region, axillary underneath the armpit, inguinal region, spleen, tonsils. MALT is mucus associated lymphoid tissue. So what they're trying to tell you is that these lymphatic tissues will actually have white blood cells that are just sitting there and waiting and monitoring fluid. And this will make a little bit more sense when I kind of explain what happens to really large items. We push them into our lymphatic rather than take them back up into blood vessels. I think this is a nice image to show you where you could find some of these. So they're trying to drive home the point that you have all of these floating in your bloodstream, and then you're going to have some in your lymphoid tissue. You're going to have macrophages. This is lung alveoli here. You're going to have those dendritic cells in your skin. And then you're also going to have mast cells in connective tissue throughout the body. So remember, any connective tissue is going to have cells, and they're going to have fibers, and it's going to have some sort of ground substance. And so we can see these mast cells, and we'll talk about the fact that they release histamines. So there's two types of immunity depending on the cells that are involved, what sort of cell response we need, how to get rid of whatever the substance is that's hurting us, right, and the amount of response time. So we're going to look at innate versus adaptive. They all kind of work together, but innate you're born with, and adaptive you have to be exposed to the pathogen for them to start working. So innate immunity, present at birth. Think of your skin, mucosal membranes where we're going to trap, dirt, pathogens, debris. Adaptive immunity, acquired, specific. You're responding to some sort of antigen. So remember, if you have a virus, viruses also have antigens on them. Antigen is something that elicits an immune response. So I know we were talking about antigens on red blood cells. Anything that's expressing a tag, a glycoprotein tag that elicits an immune response, we would call that an antigen. Adaptive, you can see this is kind of adaptive takes several days to become effective. You have to trigger cell response, trigger cell differentiation. You have to turn out tons of antibodies if we're talking about B cells. So this takes a while to be effective. Nice little flow chart here. You can see innate immunity, skin, mucous membranes. This would be macrophages, natural killer cells. You're going to have internal defenses. We'll talk about chemicals like interferon and complement. And then you have these physiological responses to stop the spread of an infection like inflammation, fever to reduce the ability of the pathogen to divide or reproduce itself, also to speed everything up in your body. So adaptive immunity, this is when we talk about T lymphocytes and B lymphocytes. And again, T lymphocytes are going to make cytotoxic T cells, and then they're also going to make helper T cells. B cells are going to create plasma cells, and plasma cells are going to generate tons and tons of antibodies. So you kind of think of this as B cells are going to make tags to tag the virus, to tag cells that are infected with the virus, and then the T cells are going to go out and kill that cell or digest that cell, right? So innate immunity, we talk about the first line and the second line of defense. So the first line is your skin, right? That's your barrier between your external and your internal environment. And then we have this second line of defense, and you can see it's a whole list, right? So we're going to stop the spread of pathogens. We have neutrophils that are, destroy bacteria. We have macrophages that are kind of scanning. They're in our blood. They're in our tissue. Dendritic cells. We have eosinophils, basophils. We have these natural killer cells. Chemicals such as interferon and complement, which we're going to talk about in just a second. And then inflammation. And so we'll talk about what inflammation is doing for us. And it says this is really, really important. So here's an example of the phagocytic activity of neutrophil and macrophages and dendritic cells. You can see the infectious agent is being engulfed, it's being broken down, and then the waste or debris is disposed of, right? Absorbed and excreted. So some of the other things that can happen, I don't have a slide, but those natural killer cells, if something is wrong with a cell, they can induce the cell to do apoptosis. So think of it as self-destruct. We also have these antimicrobial proteins, and I just wanted to show them to you because I think they're fascinating. Again, we're not going to get into all of the plasma proteins and the cytokines that are responsible for this, but interferons, you're going to see that one cell talks to a neighboring cell and prevents infection. So one cell is going to release a chemical, and we're going to see this picture in just a second, and then that chemical is going to go to the second cell, the neighboring cell, and tell it, hey, look, I just got infected, you need to up your defenses. And then we're going to talk about this complement system where we're basically poking holes in pathogens to directly destroy them and cause cell lysis. Here's interferon. We're showing you, this is from another book, so I just wanted to put this one in here because I think it's pretty simple and I kind of like it. Here's a virus. The virus enters the cell. It triggers this cell, DNA to RNA to a protein, right? And this protein binds its neighboring cell and tells it, hey, ready your defenses because there's virus in the area. So it stops a neighboring cell from being infected. This would be like one cell, when we talked about local signaling that chemicals can do, this would be an example of a local signal, one cell to the neighboring cell. Complement, we have these plasma proteins that are floating around in our blood, and these plasma proteins can be assembled if triggered, and they can poke holes into the target cell. And that therefore the water would enter the cell, the cells would lyse and burst open, and you've now destroyed the cell or the pathogen that was causing disease. It's done. We can see inflammation. This is an immediate response, right? Immediate response. It's local. There's some sort of damage, so injury to tissue, or there's an infection, and it's going to trigger these events of inflammation. And we'll look a little bit more about what exactly is happening. But remember, basophils and mast cells. So we said mast cells were in tissue. Basophils are in your blood. Both of these can trigger inflammation by releasing these chemicals. So histamines, leukotrienes, prostaglandins. This is going to trigger fever, pain receptors. The chemotaxis that we talked about a little bit with platelets, this is going to cause leukocytes or white blood cells to come to the area. So basically sending out these chemical signals that are going to cause this inflammation response. So those words highlighted again. I want to make sure that we understand what diapedesis means. So basically the cells, the white blood cells, can escape the blood vessel walls. And I'll have an image for you in just a second. If you have, let's say, infection here, right, and here's my blood vessel, I need to be able to get white blood cells to that site of infection to hopefully bind the pathogen, destroy the pathogen. And so we have to kind of create little gaps in the blood vessel. And so we call this process diapedesis. The chemotaxis, this is what I just said, where the leukocytes are going to migrate towards the release of the chemicals. So when the basophils and the mast cells are releasing these chemicals, that causes the leukocytes, the white blood cells, to migrate to that area. The delivery of plasma proteins to the site. So plasma proteins would be antibodies. This is that big word, immunoglobulins. Plasma proteins can assemble to make that complement, to poke holes. Clotting proteins in case we've had some sort of vascular damage. So I'll show you an image and it'll make a little bit more sense. The fluid that moves from the blood to the injured or infected area is going to have immune cells in it. We're going to see vasodilation of the blood vessel in order to bring more blood to the area. We're going to see that increased capillary permeability, that's the diapedesis. We're going to see a loss of plasma proteins. The plasma proteins have to get into the tissue in case they want to do the complement, right, and poke the hole in the pathogen. So plasma proteins have to go from the capillary, that's plasma proteins are in your blood, and they have to move into the injured site. So the inflammation response or inflammatory response is a little bit slow, right? We're going to see macrophages, which are digesting the bacteria. We're going to see damaged host cells. We're going to see neutrophils that are breaking up bacteria, and so those neutrophils are going to die. We're going to have to make more, right? We also have to do tissue repair. So this is a long, kind of drawn out process. That's why they call it a physiological response. There's several players here. I like this image because I think it's showing you, damage to a blood vessel, right? So bacteria are flowing into the dermis. And remember, if you have blood, if you're bleeding, that means you had to have gotten into the vascular layer of the skin, because that's where our blood vessels are. They didn't draw them, but hopefully we know that. So you can see here there's a release of inflammatory chemicals. So mast cells that are in your tissue are going to start releasing chemicals, and those chemicals are going to basically signal we have an incident, we have an issue. You're going to see vascular changes. You're going to see vasodilation. So here's what I mean by these gaps that are kind of created. So that would be diapedesis. You're going to see gaps in the blood vessels, which allows the white blood cells to move through into the damaged tissue area. And the chemicals that were released, they're either released by the mast cells, the histamines, or they can be released by the damaged tissue. So if this tissue is damaged, it's also releasing chemicals, and it recruits all these white blood cells to come to the area. Over here, we have plasma proteins. And those plasma proteins are coming out of the blood because of the diapedesis, and they're going into the tissue. So those plasma proteins, like I said, could make complement, they could poke holes in pathogens. The other thing that they're doing that's really important, that'll make a little bit more sense when we move into blood pressure and we talk more about blood vessels and we talk about lymphatic system. If you look at your blood vessel at the bottom, right, usually the blood vessel, I'm being very simplistic here, but we usually drop off nutrients and oxygen on one side. And then we pick up carbon dioxide and waste on the other side. Remember we said plasma proteins determine fluid movement in and out of blood vessels. So if I'm dropping off all of these plasma membranes, if I'm dropping off all these plasma proteins, basically I'm stopping the uptake of any of this mess. So the waste product, any of the pathogens, I'm not letting them come back into the bloodstream. And so basically what happens is they get picked up by the lymphatic system. So the lymphatic system is going to clean all of your blood before it dumps it back into your heart chamber, so your right atrium. So we'll talk about this in the next unit, but this system is really, pretty dynamic, right? We are releasing chemicals to trigger the fact that we have some sort of tissue damage or a pathogen has made it into our tissue. We're causing vascular changes, vasodilation, causing diapedesis, causing white blood cells to move out into the tissue. We're causing more white blood cells to be recruited to the area. We're dumping plasma proteins that could possibly kill some of the pathogens. And by dumping those plasma proteins, we're also stopping the uptake of all of this waste and possibly pathogens that weren't killed, right? And we're forcing all of the pathogens into our lymphatic system, where it has to be cleaned probably 100 times by lymph nodes before it actually makes it back to your heart. So this is what they mean by a physiological process. This is very entailed, makes a lot of sense. We have multiple things happening at one time. So hopefully you can look at this picture, the words make sense to you, you see the overall process, and you see why inflammation is important. Here's a list of the cardinal signs of inflammation. Hopefully we remember that blood is a little bit warmer, right? So we're going to have redness from increased blood flow to the tissue, and blood is slightly warmer than body temperature, so that's where the heat comes from, so increased blood flow. You're also going to have increased metabolic activity in that area, especially if you had damage, because you have to trigger repair, right? You're going to have swelling. Why are you going to have swelling? You're going to have plasma proteins pushing fluid in, right? So those plasma proteins pushing more and more fluid into the tissue, and you're going to have that fluid being picked up by the lymphatic system. So you have increased fluid loss from the capillaries. Those protein plasmas are directing more fluid out into that interstitial space or into the actual tissue. You're going to have pain. The stimulation of pain receptors, we talked about the prostaglandins do that. You can also think if you're doing vasodilation, there's usually nerves close to blood vessels. So if you're doing vasodilation, it's pushing on those pain receptors. We also have a loss of function, right? So the pain and the swelling in severe cases is going to limit your ability to move, right, or function in that particular area. And this is actually kind of a good thing, right? Basically, we've got a lot of cleanup to do. We've got things we need to take care of. You need to stop using this particular area. So acute inflammation, 8 to 10 days. If you have chronic inflammation, I'm having trouble talking this morning. This has detrimental effects, right? Long-term inflammation of tissue can cause damage to the tissue, damage to the blood vessels. You can see here the benefits of fever. It's actually having a slightly elevated fever is good for healing. So you can see that it inhibits the reproduction of bacteria and virus. There's something about the sequestering of zinc, some minerals that are needed for bacteria to copy themselves and reproduce. And so having a fever kind of limits the materials that they need in order to reproduce. It can promote the interferon activity. So increased body temperature kind of triggers that one cell to release interferon. There's bacteria in the area, there's infection in the area to alert other cells. It can increase the adaptive immunity, which is what we're getting ready to talk about. So hopefully when you see the term adaptive immunity, you're thinking T cells and B cells. Having an elevated fever can actually accelerate tissue repair. It can call more immune cells out of the blood, act as a target. So a low-grade fever is okay. We probably don't want to treat it. Let it run its course. It becomes dangerous if your internal body temperature goes above 104. This is when we're breaking down good enzymes, right? So think about the enzymes necessary for cellular respiration. So if I need to turn glucose, I'm not writing the whole equation here. If I need to take glucose and turn it into ATP, and we talked about the fact that there are several enzymes responsible for this process. Imagine if your good enzymes stop working, then you make less ATP. Now you can't, run all of your muscles efficiently. Your brain activity not as efficient. So a little bit of a fever, okay. You're probably doing good. If the fever goes out of control and over 104, 105, then we need to worry about it. So now we get to the adaptive immunity. Hopefully when we think about adaptive immunity, we're thinking about T lymphocytes and B lymphocytes. You'll hear me just say T cells and B cells. So they call it cell-mediated immunity, and they have antibody-mediated immunity. This takes a little bit of time to develop. So if you've ever been sick, your nose is going crazy, right? You're all stuffy, you don't feel good. And then it takes probably, 6-7 days. And then all of a sudden you're like, oh, I feel better, right? Well, that was enough time for this adaptive immunity to kick in. We can see T cells. They're going to divide into cytotoxic and helper cells. So I'll call them cytotoxic T cells and helper T cells. And then we have B lymphocytes or B cells. They're going to make plasma cells, and those plasma cells are going to produce antibodies. So the helper T cells, again, we're not going to have to know all of these different receptors. So if you take microbiology, you will. If you take an immune class, you'll have to know a little bit more. Right now, I just want you to know that the subtypes of T lymphocytes or T cells, we have helper T cells, we have cytotoxic T cells, and we have something called antigen-presenting cells, which does exactly what you think it does. So it sees an antigen, and it brings it to your immune system, and it says, good or bad, right? Is this a good or a bad antigen? Is it a self-antigen, or is it a pathogen that I need to get rid of? They're showing you here the helper T cells, and I kind of like there's a larger image that I like a little bit better. So I'm going to skip through these and come to this image. So this is from another textbook. I think it kind of shows the picture of everything happening at once rather than individual. So here we have, get my pen working. Here we have our immature lymphocyte, right? So remember, we have to go from hemocytoblast to get to like a midway point, and then we're going to have this maturation. This happens in the thymus. You have two different cells. This is what I meant by, they call them CD4 cells and CD8 cells based on these receptors. But you can see we're eventually going to make helper cells, and then down here we're eventually going to make cytotoxic T cells. So your immune system gets educated. We'll talk a little bit more about that through other chapters, but basically this is an antigen presenting cell. That's what APC means. So this cell picked up an antigen and it says, hey, I found this, and it brings it to the T cell, and if it's a pathogen, it will activate this differentiation and you'll make cytotoxic T cells. And they're specific to this antigen. So this receptor is specific to this antigen so that these cells can bind and destroy pathogens with that tag. Pathogens with that specific antigen. They're also going to create these helper T cells. Activation of B cells. So B cells are going to make antibodies, and they're going to differentiate into the plasma cells. The remainder become memory B cells. So basically, you're going to have a B cell, and that B cell is going to differentiate into 3 plasma cells. They're going to make antibodies, and this one is going to become a memory cell. And what triggers the B cell to do this process? There has to be an antigen that triggers that B cell, because what are we making? We're making antibodies. What do the antibodies have to be specific for? They have to specifically bind the antigen that's foreign, that's not supposed to be there, right? So another example here, they're showing you the B cell, and the fact that this B cell is going to differentiate into a memory B cell, and it's going to activate these plasma cells, and the plasma cells are the ones that are actually producing the antibody. The memory cell basically is there as a memory of infection. You'll be able to make antibodies more quickly if you have these memory B cells, if you're infected with the same antigen. So I also want you to see the helper T cell over here. So it does just that, right? The helper T cell can help activate the B cell into producing antibodies. So we have cell-mediated immunity. This is T lymphocytes. We have helper T cells. We have cytotoxic T cells. And then we have antibody-mediated immunity, and that involves B cells, which are going to become memory cells, and then plasma cells, and the plasma cells actually produce the antibodies. So think of the helper T cells as kind of activating, right? They can help activate B cells. They can help activate cytotoxic T cells. They can go back and stimulate your innate, keep the inflammation going, keep the fever going. So they kind of just are all around helpers in making sure that your immune system keeps going and keeps fighting the pathogen until it's all gone. I listed all these immunoglobulins, or these IGs. You don't have to know them. I just want you to see that there's different classes of antibodies, and they're produced against particular antigens, and you're going to find them in particular places. So just so that you can see it, it's not something that you'd be tested on. So the reason that we have those memory cells, those B cells, is because it produces this powerful secondary response in a much shorter period of time. So what'll happen is that memory B cell can be activated much more quickly. So you have that memory B cell, and instead of going through the long process of making the 4 and the plasma cells and the memory cell and being activated, basically just this memory cell is activated and you start churning out plasma cells, which then make antibodies. So it's a much quicker, rapid response the second time. The only problem is it has to be the exact same antigen. So if it makes contact with the same antigen, that memory B cell can start churning out tons of antibodies. So usually the pathogen, if it's a second response, the pathogen is usually eliminated before disease occurs. You know, kind of funny sometimes if you're not feeling really well, you feel kind of tired and slack, you feel like you're fighting something. I've said that so many times, man, I don't feel bad, I don't feel great, I feel like I'm fighting something. My immune system has probably been exposed to an antigen that I've seen before. My memory B cells are overactive, churning out tons of antibodies, and I just feel kind of sluggish, right? And then the next day you feel fine, or the day after that you feel, you know, back to normal. So that's that secondary response working for us. So we don't have to start from the beginning of the process every single time. I wanted to mention vaccines. A lot of us, you know, are probably a little bit more familiar with vaccines due to COVID. So if I give you a vaccine, it's a weakened or it's a dead microorganism or the component of. So what they're saying is if you have a virus and you have this antigen, I'm going to give you just this piece in the vaccine. So if I inject you with a bunch of antigen from a virus, your immune system is going to recognize it. Your immune system is going to start producing antibodies, but you've never been exposed to the inner workings of the virus, like the DNA or RNA. So the virus can't copy itself. All we're giving it is the tag. So people that say, you know, I got sick, you know, every time I get a vaccine, I get the disease. That's probably not true, right? They either had the virus already or they're allergic to some other component, but you cannot get the virus from just getting the tag, right? Herd immunity, another term that we've heard a lot about. Basically, you have to have a really high proportion of individuals that are vaccinated in order to keep the disease at a minimum so that you don't see the disease anymore in the population or with as great numbers as you did. I wanted to put this up here. I love Twitter. I love some of these medical things that come up. I follow a bunch of Twitter medical pages, and I loved this image. I think this is just amazing. And so I've given credit at the bottom. It's a little bit older, you can see from 17, but I think it does a fabulous job of showing us that vaccines really do work. So this is over a long period of time. This is back in 1945 all the way to present day, and or until they did this article, and they're showing you that when we had the vaccine, we see a dramatic drop off in infections, right? So they're showing you each of the diseases here. The yellow circle is when we had the vaccine licensed. This is before, this is after. And you can see, I mean, just from this image, we can really see how well vaccines work if enough of the population is vaccinated. You can also follow this pertussis, and we're starting to see a little bit more of an outbreak here. Think about whooping cough. That's kind of the DPT, the pertussis part of it. So we have to be careful, right? Vaccines are good. Vaccines do prevent infection. But we can't be lazy, right? We have to make sure that we stay vaccinated, that children, when they're born, stay vaccinated if we want to keep these diseases to a minimum. The last slide here is just to really talk about passive versus active immunity. I wanted to make sure that active immunity is when we trigger T cells and B cells, right? So active immunity has to be triggered by an antigen. So can you be exposed to the antigen and trigger production of T cells and B cells? Yes, so you got sick. So this is, I got sick, I contracted a virus, and I made T cells and B cells against it. You can have artificially acquired. So this is our vaccine. I gave you the outside glycoprotein. I triggered your immune system to make T cells and B cells. And then over here we have passive immunity. So you do not make memory cells. You did not trigger your adaptive T cells and B cells. So naturally acquired. So across the placenta or in breast milk, we can transfer antibodies from mother to child, right? The only thing is antibodies don't last forever. So as long as the baby is getting more and more antibodies from mom, everything's fine. As soon as mom stops breastfeeding, no new antibodies, the child doesn't have the memory cells to make those antibodies, right? We never triggered the active immunity. If you've had a child, you know that about a year after, when they get off of breast milk, if you take them to daycare or they do play dates, then all of a sudden they're sick all the time, right? So they're now building their adaptive immunity. They're being exposed, they're getting sick, and now they'll have those memory cells. But just that first year of life, they're getting them from mom. Their immune system is not highly developed. We also have artificial passive immunity. So what if I gave you serum from somebody that had antibodies? I can give you those immunoglobulins, but again, they're artificial, they're antibodies generated synthetically or from other animals, and eventually it's going to break down. You're not going to have the memory B cells. So think of, passive immunity is kind of a short-term solution. You're not creating the lifelong active immunity of T cells and B cells. Last slide here is to talk about HIV. So AIDS is acquired immunodeficiency syndrome. It's a result of human immunodeficiency virus. This virus infects and destroys helper T cells. So remember how important these helper T cells were. They go back and they tell the innate to keep going, right? The inflammation, the fever. They can recruit white blood cells, right? They can tell cytotoxic T cells to keep going. They can go back and tell B cells to make antibodies. So several individuals, when they passed away or died from HIV, that wasn't the infection that was listed. Usually it was, the person had pneumonia, they had TB, right? So the cause of death is usually something else, some disease that their immune system could no longer fight because HIV destroyed all of their helper T lymphocytes. So it kind of shows you how important those helper cells are. Even though they're not doing the main job, they're kind of going back and telling everybody to keep going. All right, well, that is it for chapter 22. I know there were some complicated things in here with inflammation and some of the creation of those T cells and B cells. Can you look at the picture? Do the words make sense to you? If somebody asked you to explain inflammation, could you explain all the parts of inflammation? There'll be a few questions about this chapter, but basically stick to the overview. That's what I'm asking you. Anything that was in this PowerPoint, anything that's on your learning objectives, that's what you need to focus on. The rest of the chapter, you can read it and enjoy it, but it won't be part of the test. So I'll talk to you guys again later. Bye.